Hi, welcome back. So this video covers blood vessels and blood circulation, and this is all in chapter 20 in your book. So you're going to want to read chapter 20 and reference that along with this lecture. When we talk about blood vessels, we sort of divide them into three primary types. We have arteries, which are going to carry blood away from the heart toward the capillaries. We have tiny, tiny blood vessels, microscopic. They're usually only one cell layer thick, called the capillaries, and that is where all the exchange happens. And then from capillaries, blood will flow back into veins and go back to the heart, and then we'll start over again. So let's think about this general pathway kind of real fast. So we think about heart. From the heart, leave the heart via arteries. Those arteries will branch and branch and branch into smaller vessels called arterioles. From an arteriole, we will go through a capillary bed, and the capillary bed is just a collection of a bunch of capillaries. From a capillary bed, we go out into smaller veins called venules, finally into actual veins, and then back to the heart. And you guys know this when we've talked about the heart. So in general, blood vessels are made up of layers called tunics, and then we call the space inside the lumen. Remember, a blood vessel is like a water hose, like a pipe. So now, from lab, and then in section 20.1a of your book, you're going to want to look back and make sure you can describe the structure and the function of the three tunics of the blood vessels. Paying special attention to the tunica media. So arteries and veins, and you'll see this when you do your blood vessel lab, you usually will have an artery beside a vein. They'll supply the same area. They usually have the same name, except you'll call one, say, the brachial artery and one the brachial vein. So we've got some differences between arteries and veins. So arteries have a thicker tunica media, or a thicker muscular wall. Okay. Veins have a bigger lumen. And veins have valves. So take a look at these two vessels here side by side. We're going to call them A. We're going to call them B. So take a look at these. You might need to pause your video, and then I'm going to ask you to answer a question about which one of these you think is the artery and which one of these you think is the vein. So this vessel here is our vein. Right here is the tunica media, or that muscular layer. You can see it's really thin compared to the smooth muscle layer in the artery. And again, this vein's got this big, wide lumen. There's some erythrocytes kind of clustered in there, versus the artery, which has a much narrower lumen because of all that smooth muscle. And then we don't see any valves in this image. So one of the things we're going to need to review is what kind of blood arteries and veins carry. So remember, arteries carry blood away from the heart, and veins carry blood toward the heart. And so now let's go back, answer a few questions about which arteries and which veins carry oxygenated versus deoxygenated blood. So our arteries after they leave the heart, they're going to progress into smaller and smaller vessels. As they get smaller, the lumen diameter changes because they're smaller, and the composition of the wall changes. And so we'll start near the heart with our elastic arteries. That would be something like the aorta, and they've just got a ton of elastic fibers in their wall. So that allows them to kind of stretch and expand as blood is pumped out of the heart. All the way down to arterioles, which are then very, very small. And so the kind of the progression of it is when we just come out of the heart, we have elastic arteries. Get a little further from the heart, they branch and give us muscular arteries. Muscular arteries branch to arterioles. And each arteriole is going to supply usually a certain region of the body. So this is another independent thing that you don't need to listen to me talk about. You can pull right up from your book. So just be sure you understand the difference between elastic arteries and muscular arteries. And you especially want to think about what are the elastic arteries, and specifically what are they? And they're not many. They're the ones that are very close to the heart. So be sure you make a list of those. What we want to talk about is arterioles. So they are the smallest but the most abundant of the arteries. They don't have a ton of smooth muscle, and they sit around with their smooth muscle and sort of a little bit constricted. So they're never completely relaxed. Because there are so many of these things, okay, they have some really important functions. So one of them is this is really what your body does to direct blood flow. What I mean by that is maybe you just ate a big meal and you're trying to digest, so your body will want to send more blood to your GI tract to absorb all that stuff. And so the artery supplying the GI tract might dilate. And other times arteries too, like the skin would constrict when you were cold. So they help with blood flow, they help with body temperature, and they really help with maintenance of blood pressure. And I know, I swear, through this whole unit, I've just talked and talked and talked about blood pressure, and we're going to talk about what that really means very soon. But just keep this with our arterioles in the back of our minds. As you hopefully learned in Bio 169, arterioles are controlled only by the sympathetic division. And they are controlled, that control kind of starts in the vasomotor center, which is in the medulla oblongata. You should never write medulla without oblongata because there's a few other medulla structures in the body. So that was my bad. So the way these arterioles work, there's a baseline of sympathetic stimulation. So they're always getting kind of a certain amount of sympathetic stimulation. Okay. We call this vasomotor tone. Anytime, guys, we see vaso, from now on, we're going to think having something to do with a vessel. Okay. And then we think about motor means movement, but really that has to do with the vessels constricting or relaxing. Okay. So, as a general rule, over the entire body, if your sympathetic stimulation increases, we will have overall in the body net vasoconstriction. Now, there are exceptions to this, but the vast majority of vessels in your body will constrict in response to sympathetic stimulation. Okay. The ones that don't are the ones that are supplying skeletal muscle and the heart muscle. Okay. But again, the vast majority of the vessels in your body are the vessels that are going to your skin and to your organs. Okay, so they're going to constrict in response to the sympathetic nervous system. And then if the sympathetic nervous system stimulation decreases, then we're going to get what we call overall in the whole body a net vasodilation. So I give an example. This is like the gas pedal. So you want to go down the road at 60 miles an hour. You've got gas, but you don't have a brake, okay? So you're 60 mile an hour holding your foot on the gas, that's your vasomotor tone. If you want to go faster, you push the gas, right, and you'll go faster. If you want to slow down, you don't have a brake, so you'll let your foot off the gas, and that's how these arterioles work. Capillaries then are the tiny, tiny blood vessels. They have only a tunica intima, which is that inner tunic, and it is a simple squamous epithelium, which means it is one layer of flat cells. Very easy to cross. And so that structure makes these capillaries really good at exchange, because what happens in the capillaries is we move things out of the capillaries out to the interstitial fluid so the cells can access them, and then things move from the cells into the capillaries. Now they're in the bloodstream and then go back to the heart. There are a few different structures of capillaries based on how easy they are to cross and what sort of things can cross them. So, brings us to another independent adventure. So in section 20.1c, we talk about the 3 different types of capillaries. Be able to describe them and describe what kinds of things can cross them and some examples of where they're found. For now, right now, we're mainly going to be talking about the continuous capillaries. We'll talk about the fenestrated capillaries with the kidney mostly, and then the sinusoids are mainly in the spleen and the bone marrow. Okay, but right now we're mainly going to talk about continuous capillaries because that's what most of the capillaries in your body are. So, capillaries, we think of them as arranged in these structures called capillary beds, and that is a big group of capillaries. They function together and they supply a certain structure. Okay, so it's going to look like a big, I think of it as like a bunch of lace, like a big lace overlay of all these little interwoven capillaries supplying an area. So this is sort of how they work. We've got an arteriole, one of those itty bitty arteries, which is going to bring blood to the capillary bed. It's going to branch, and we'll look at the meta-arteriole in just a minute. We have a thoroughfare channel that's going to pass through the capillary bed. After we've passed through the capillaries, we enter into a venule, which is just a small vein, and then a bigger venule that's going to drain the capillary bed. And when I say drain, I just mean blood goes into the capillary bed, leaves the capillary bed via this venule, and now it's on its way back to the heart. So then we have these things called true capillaries. True capillaries are going to branch off this stuff up here, and this makes no sense right now, I know, but we're going to look at a picture next. At the opening of each capillary is a ring of smooth muscle called a precapillary sphincter. Let's look at that and it will make more sense. So here is, I'm going to change colors because there's a lot of purple going on in here. Let's do green. So here is a capillary bed. Okay. We've got an arteriole bringing blood toward the capillary bed. The meta-arteriole is just this little area right here that's going to kind of branch off toward the capillary bed. We've got a thoroughfare channel here, and a thoroughfare channel is just a road that passes straight through the capillary bed. Okay. From off that thoroughfare channel, then branch all these actual capillaries. Okay, so these are your actual capillaries here. They've got little sphincters that close them off. Okay. Blood goes through here, and now look, this blood started out red over here. It's purple here, which means that gas exchange is happening. Okay, so remember, we show oxygenated blood as red and deoxygenated blood as blue in our books, and so purple means we're leaving oxygen as we cross this capillary bed, and then leaves the capillary bed through this postcapillary venule and into another venule, which again is just a small vein. Okay, so this is what we think of when we think about a capillary bed. Now, this thoroughfare channel is interesting because you do not have enough blood in your body to fill all your capillaries at the same time. So at any given time, some of them are closed. So those precapillary sphincters just kind of seal down. And rather than flow into these capillaries and do a ton of exchange, blood just goes straight through from the artery to the vein. Okay. So again, keep this in your brain. Maybe to go back and look at the picture in your book. It's got a lot of words, but we're really just thinking little artery, capillary, little vein. So when that precapillary sphincter is relaxed, blood is going to go into the true capillary. I'm going to abbreviate it CAP. Okay. And when that sphincter is contracted, blood is just going to pass through the capillary bed via that thoroughfare channel. Those sphincters have a cyclical pattern of contraction and relaxation. So they contract, relax cycle happens about 5 to 10 times a minute. That is called vasomotion. Now, at any given time point, only about a quarter of your capillary beds are actually open and filled with blood. So there are 60,000, which this is just like an absurd number to me, but there are 60,000 miles of capillaries in your body. And at any given time, you've only got a little bit of blood, okay, in the capillaries because the rest of it's outflowing through the arteries and veins, etc. So there's just not enough blood to fill all your capillaries. Okay? So perfusion, which we've talked about before, is kind of adequate blood supply to a tissue, and that directly has to do with capillary beds. Okay, and perfusion, we can also sort of measure it in amount of blood, usually in mils, per gram of tissue per unit of time. Okay, but it's just really a measure of how much blood is this specific tissue getting. And if a capillary bed's all closed off, that tissue is not getting good perfusion at the moment. That's fine. That's how your body's set up. So once we leave the capillary beds, and we will be back for the capillary beds because we're going to take a deep dive into capillary exchange, we enter into veins. Okay? Veins are going to carry blood from the capillary beds or capillaries back to the heart. So when we talk about veins, we talk about them as merging and coming together. So for example, let's take your hand. You got a bunch of tiny veins in your hand. They'll run together into bigger veins that are in your forearm, and those will run into bigger veins that are in your upper arm. And those will merge together into bigger and bigger veins. When we talked about arteries, we talked about them splitting, and that just has to do with the direction they're going from the heart. Okay, so they're just an anatomic structure, but we just talk about veins as merging to form larger and larger vessels. Veins have lots and lots of valves. By the time your blood gets to the veins, there's not a lot of the heart's pressure left to push it. So we have valves in there for that one-way flow of blood so that we keep blood moving toward the heart and it doesn't settle and pool. So that's our arteries, our capillaries, and our veins. When we are at rest, this is how our blood is distributed throughout our body. So you got about 20% of your blood in the pulmonary circulation, the rest of it in the systemic circulation, about 10% of it is actually in the heart. So if I hit pause on you and everything stopped and we measured your blood, this is where it would all be. Of that systemic circulation, there's a little bit in the systemic arteries, a little bit in capillaries, but at any given time, over half of your blood volume is actually sitting in the systemic veins. Again, so if I hit pause on you right now, measured the blood in all your vessels, over half your blood is in your systemic veins. What that does is allow those veins to act as a blood reservoir. Because there's a relatively large volume of blood in those veins. Okay. So when needed, so if you are exercising, blood can be shifted from those veins so you can put a greater percentage of your blood into the rest of the circulation. Okay. So that really happens by a couple ways. So there is something called venoconstriction. So the veins actually constrict and get smaller, and therefore they don't have enough room for blood. Also, skeletal muscle contraction can squeeze those veins, especially out in your arms and legs. Push blood along toward the heart. This is mostly happening when you are active or not at rest, because your tissues, especially your muscles, all need more blood to meet their energy demands. All right, so now we've gotten kind of familiar with the kinds of vessels we have. We're going to move into talking about this concept of total cross-sectional area and then blood flow velocity. Again, velocity, guys, is basically speed, okay? So it's the amount of blood per unit time, okay? So blood flow velocity will change, but blood always goes along, or you could say down, a pressure gradient. And pressure is how hard something's pushing on its container. So if given a choice, a liquid or like blood wants to go from high pressure to low pressure. So total cross-sectional area, I'm going to read you this definition, then I'm going to explain it. The aggregate lumen diameter across the total number of a specific type of vessel. So think about it as you got a certain amount of space inside your blood vessels, okay? How much space across your body is in the arteries? How much space of your total vessel space is inside capillaries? Okay, so that's what this total cross-sectional area is, just how much space is there available in all the arteries together in your body. And so we look at it with a little graph, which is kind of weird. So this is vessel type. So here is the different kinds of vessels. So these big elastic arteries all the way back to the vena cava. So we're moving away. Here's the heart. You know, we're moving, blood is going this way, and then it'll come back to the heart. So let's look. So these are what kind of arteries. Here is the total cross-sectional area. And then here is how fast that blood is going. That's our red line. So we can see that here in the elastic arteries, blood's going really fast, and as it goes to the capillaries, it really slows down, speeds back up a little as it gets into the veins. So that is how fast. Cross-sectional area, and again, that's how much space in your vessels are in a certain kind of vessel. And you can see that even though capillaries are teeny, teeny, tiny, you have so many of them that most of the total cross-sectional area of your vessels, most of the space in your blood vessels, is actually contained within those capillaries. So even though these elastic arteries or something like the big vein like the vena cava are huge, there's only one of them. So there's not much, again, across all your body, not much of your vessel space is inside an elastic artery or a big vein. So let's talk through in a little more detail what this graph is. So let's work through that graph in a little more detail. So look at our graph. Which group of vessels, so your answer is going to be something from down in here, has the greatest total cross-sectional area? So stop here, take a moment, maybe you need to pause the video, look at this graph, okay, and then go and answer the question, and I'll meet you back here. So capillaries have the greatest total cross-sectional area, which means most of the space inside your vessels is within the capillaries. Does that make sense, given the anatomy of the blood vessels? I think yes. And so you're going to think yes because I told you to, right? The reason that makes sense is because you have many capillaries. Over 60,000 miles of them. Okay, so of course, most of the space in your blood vessels is within the capillaries. So what is this graph telling us? And another thing it tells us is the relationship between total cross-sectional area and how fast blood can flow. So take a moment, again, pause if you need to, and then answer the question and tell me the relationship between total cross-sectional area and blood velocity. So what is that relationship? So that relationship, let's look. Here in our big elastic arteries, area is very low, velocity is very high. Here in our capillaries, lots and lots of area, very slow velocity. And then here in the veins, as our area goes down, velocity picks back up. So I would say as total cross-sectional area increases, blood velocity decreases. So what's going on here? So the capillary beds, again, they're tiny vessels, and there's tons of them. Blood just goes slower through those tiny vessels. I would think about it like, think about your big elastic arteries as I-40 with, 4 lanes on either side, right? And you've got 40 cars going down I-40. They're going to go a lot faster and get to their destination a lot faster than if I put them on 40 different 2-lane winding roads, right? They'll have to go a lot slower. So that's kind of what's going on there. These capillaries are little winding roads as opposed to the I-40 that is the elastic artery. So what does that mean for us? Blood goes really slow in the capillaries. Okay. Why do we think blood has to go very slowly through the capillaries? There's an important physiologic reason that happens. And so we'll see if you can answer the question and tell me why. And next time we're going to talk about exchange and what actually happens in the capillaries.